So our first speaker this morning will be Dr. Liu Chen. So Dr. Chen?
Yes, yeah.
Are you going to give us some welcoming and introduction address?
Yes.
So Dr. Chen is the Associate Director of State Key Laboratory of Marine Pollution and a visiting Associate Professor from Department of Biomedical Sciences, CTU. So he has very broad interest in ecology and biodiversity of marine ecosystem. So Dr. Liu Chen will give us some welcoming address.
Yeah, thank you, Harry.
Yeah.
So on behalf of the State Key Laboratory of Marine Pollution at the CTU University of Hong Kong, I would like to welcome all of you to participate today's symposium. So as you know that yesterday we have a training workshop, following by today's one-day symposium, which will feature a series of talks on recent advances in marine environmental research, including the latest technology of pollution monitoring and control, occurrence and impact of emerging chemical concerns, problem and imminent stressors such as hydroxia and algal toxins, and also novel approach in ecological restoration. Particular attention will be placed on toxin and man-made substance that pollute the ocean or affect its living biodiversity. Participants will have a chance to express their view on the challenging marine environmental research during the panel discussion chaired by Professor Rudolph Wu. So Henry already introduced me. I'm the Associate Director and also the Director of Shenzhen Key Laboratory for the Sustainable Use of Marine Biodiversity. As Rudolph is here, the State Key Lab is built upon the excellent leadership of Professor Rudolph Wu, established the Area of Excellence Center of Marine Environmental Research and Innovation Technology in 2004. And then Rudolph is continue taking the lead in organized PEMSEA Regional Center for Excellence in Marine Pollution over the last, I think, 12 years, Rudolph, right? And the State Key Laboratory, the major reason is to be a key international research center in advancing marine environmental research that contribute to the protection and management of the marine environment and generate positive societal impacts. We have three major missions to protect marine environments of Hong Kong, South China, and Asia Pacific regions. So that's why we organize the training workshop and today's symposium for the purpose of putting more interdisciplinary expertise through high quality multidisciplinary research and innovation relevant to pollution monitoring and control, environmental risk assessment, ecosystem response to stressors, and ecological restoration, and also to build capacity by nurturing and training environmental scientists through yesterday's workshop and today's symposium, and manager and industrial entrepreneur in the regions, and also to support Hong Kong SR government and the Chinese central government in the management of environmental quality and protection of marine ecosystems. So the uniqueness of MERI SKMP is its inter-university, multidisciplinary, inclusive, always emphasized by Professor Wu to pull together all the expertise in Hong Kong to conduct collaborative research that gather together many minds to become greater together, to be inclusive, not exclusive, to tackle grand challenges, create impacts together. So we have three strategic research fields. The first one is led by Professor Zhang Tong, the first speaker, and then Dr. Guo Zhenji, and he will give a presentation in the later morning. And Professor Wang Wenxiong lead the second strategic research field, eco safety and environmental risk assessment. And Dr. Henry He to be the deputy leader for this direction. And also Professor Chu Jianwen is also here leading the ecosystem response and ecological restoration with my assistance. So there are two pieces of good news from SKMP. SKMP will lead UN endorsed project, the UN Decade of Ocean Science for Sustainable Development Framework, proposed by Professor Kenny Wu, the director. And also we are in application a regional training center on pollution assessment and monitoring. So thank you all for your attention, and I pass to Henry for chairing the following sessions. Thank you.
Yeah, thank Leo. Yeah, thank you very much. So now we are just going to start our first morning session. So the first speaker will be Professor Tong Zhang. So Professor Zhang is a chair professor in water and environmental engineering from Department of Civil Engineering, University of Hong Kong. His main research areas include wastewater engineering and biological treatment, anaerobic digestion, environmental microbiology and biotechnology, environmental bioinformatics, and environmental toxicology related to wastewater treatment. Today, he will present mechagenomic insights into antibiotic-resistant genes in environments. So Professor Zhang, the podium is yours.
Thank you, Henry. Thank you for kind introduction. Hello, everyone. I'm Zhang Tong from the Department of Civil Engineering, the University of Hong Kong. My laboratory mainly works on environmental microbiome. And since 2007, we have done some research about antibiotics, the chemical pollutants, and the antibiotic-resistant genes, the biological pollutants. Today, it is my great pleasure to have this opportunity to share with you our study on ARGs using metagenomic measure. As you may know, in 2015, the World Health Organization, WHO, listed antibiotic resistance as one of the biggest challenges facing human health in this century. According to a report published by ONU in 2016, by 2050, if no action taken, the problem of antibiotic resistance cannot be effectively solved. 10 million people will die due to the diseases as related to drug resistance every year, exceeding the deaths due to the cancer. Antibiotic resistance, of course, is a medical issue first, but it is not only a medical issue. It also an issue having environmental dimension. According to the widely accepted scientific concept, One Health, the health of human, animal, and environment are closely connected. Antibiotics used by human account for 30%, and the remaining 70% is used by animal. After use, 80% of the used antibiotics will be discharged into the environment. At the kind of the selective pressure, it will become the important factors in promoting the spread of resistant genes in the environment. At the same time, the drug-resistant bacteria and the other resistant genes produced in the course of different use will enter the environment with the discharge of sewage and pollutants and become the main sources of the resistance in the environment. The environment is the key node in the spread of antibiotic resistance. Bacteria in soil, river, lake, and seawater could develop resistance due to the drug-resistant bacteria, antibiotics, and other chemical substances used by human beings, such as disinfectant, heavy metal, and so on. Human and livestock will be infected by more drug-resistant pathogens through food, water, and air contact. And the antibiotics used in response to these infections will begin another new round of the drug resistance evolution cycle. United Nations the environment, United Nations Environment published the Frontier Report 2017 and pointed out that antibiotic and resistant genes have become environmental issues of serious concern. There are different measures to analyze the resistant genes in the environment. The basic method is to isolate the bacteria on selective media and then determine whether they carry resistant genes or not based on the phenotype. That means which antibiotics they are resistant to. Molecular methods such as PCR and sequencing have been used to study the genotype of resistant bacteria for quite a long time. And the quantitative PCR can detect the concentration of resistant genes in different environments. If we can design suitable primer and probe, then we can use high throughput quantitative PCR or DNA chips to study hundreds of different resistant genes at the same time. With the rapid development of second and third generation high throughput DNA sequencing technology, metagenome methods are becoming a widely accepted broad spectrum quantitative detection method. Of course, there are other methods also could be used to study ARGs together with the above method. Compared with the metagenomics, the throughput of qPCR method is low since the number of available primers are limited. Additionally, qPCR methods are affected by many factors, including the reaction system, qPCR instrument, the matrix effect, and the training of the operators. It is very difficult to standardize since the resistant genes are a global issue. Scientific research and environmental management need to have more systematic and comparable data set to identify the important pollution sources and compare the overall level of resistance in different countries and regions. This requires a relatively standardized analytical method. At this stage, the metagenomic method as a method with a high throughput, more standardization, and also better openness has been applied more widely. It not only can analyze the known resistant genes, but also may help us discover new types of resistant genes if we combine it with the functional screening. More importantly, the unknown resistant genes in the previous DNA sequence dataset could be retrospectively rechecked for reanalysis and reminding when we have knowledge of the novel resistant genes. In terms of the detection sensitivity and customization, the qPCR method and still it's much better than the metagenome method. So in summary, currently the qPCR method and the metagenome method have their own strengths and complement with each other. But to study the resistome, that means the whole collection of antibiotic resistance genes in a complex environmental system, the metagenome method is much more preferred considering the above benefit, which I mentioned. The database is the core of the metagenomic detection method. The completeness of the database will significantly affect the accuracy of the detection result. If the database is not that complete, the abundance and the diversity of resistant genes in a sample will be underestimated. So the key technical bottleneck of metagenome method is how to effectively and accurately expand the existing resistant database. At the beginning, we merged two widely used resistant gene database and then produced a more comprehensive one. Based on that, we made some improvement and developed the online ARG-OEP analysis platform for metagenomic analysis using the second generation sequencing dataset. This analysis platform could be used to comprehensively analyze the resistant genes in environment sample. It can simultaneously detect 1,200 resistant subtypes, cover 24 types of antibiotics. In the second version of this database, we add hidden Markov model for to perform high throughput screening of new resistant genes. Currently, this database and the platform become a widely used tool for the analysis of environment antibiotic-resistant genes. The website of our tool had been visited for more than 25,000 times up now. Using the established metagenome measure, we analyze the resistant genes of 10 types of typical environment samples and study the entire chain of the water environment system, including the sediment, soil, river water, drinking water, sewage treatment sludge, human gut and sewage influent, livestock manure, and farm wastewater treatment system. The total abundance of resistant genes in different environments is closely related to the degree of human influence. From the natural environment to the livestock, the total abundance of resistant genes has increased by 3 orders of magnitude. The main resistant genes include aminoglycoside, beta-lactam, quinolone, sulfonamides, and the tetracycline resistant genes, which are highly consistent with the type of widely used antibiotics, showing that antibiotic use affects the distribution of the resistant genes in the environment as the major driver of spread. The influence of the sewage treatment plant could be used as the representative samples of resistance in the gut of human population of 1,000 people from that catchment area. Wastewater treatment plant is an important ARG source in the environment, especially the widely used activated sludge since 1950s. Due to adsorption, antibiotics enriched on the surface of sludge floc could be a significant selective pressure, plus other factors such as high bacterial diversity, higher biomass, short distance between the cells, continued mixing, providing a very favorable condition for the horizontal gene transfer of ART, so-called HDT. This ecological environment is completely artificially created and developed in the recent 60 years. It does not exist in a natural environment. The average generation time of bacteria in active sludge is about 15 to 30 days, which is much faster than the growth rate of bacteria in natural water and soil. This means in active sludge, the enhanced horizontal gene transfer, HGT, together with rapid growth of bacteria, will further enlarge the impact. At the end, the resistant bacteria will be discharged into the environment together with treated wastewater. 1 cubic meter treated wastewater usually carries up to 1 billion total bacterial cells. The continued discharge of a large amount of wastewater definitely will increase the resistance in water and sediment. In addition to the snapshot sample from different environments, long-term sampling tests of the sample of typical environment are also very important, because only in this way we could fully understand the spread and evolution of resistant genes a long time. So far, there are limited studies based on long-term sampling of active sludge. We have conducted sampling for 14 consecutive years monthly at the wastewater treatment plant of Hong Kong. And here are the results of 9 years from 2007 to 2015. The results showed that the abundance and the profile of ART changed significantly every 2 to 3 years. Additionally, compared with the soil resistance, the resistance of active sludge had higher horizontal gene transfer potential. Although wastewater treatment plants are considered as critical points in disseminating ARGs, there is a limited study about assessment of the resistance and the mobilome in effluent in a quantitative way. we proposed an absolute quantification metagenomic method by considering the mass of sequenced DNA and also the total mass of extract DNA from a sample. Of course, we assume 100% extraction efficiency and to assess the resistant and also mobile genetic elements in the effluent in the cities of North America, South America, Europe, and Asia. The metagenomic absolute quantification results are comparable to those obtained using qPCR. We propose a risk ranking system based on ARG abundance in the unit of copy of ARG per cell. Hong Kong wastewater effluents were ranked in the moderate level, that means level 2 and level 3. Using the same method, we established a public platform. Actually, it's an interactive map for researchers and the government officers, all the general public, to search online for the information of the risk system in the wastewater treatment plants of different cities, and to understand the potential risk level based on the global metagenomic dataset of ARG. Here is an example, shows the ARG profile in the city of Japan. Analyzing the occurrence and the types of resistant genes in different environments is only the first step. More importantly, we must understand its dissemination and HDT mechanisms in the environment. This requires answer to a series of questions, including the host of the resistant genes, the associated mobile genetic element, MGE, and so on. To obtain this information, short sequences are not enough. We need advanced metagenomic measure using long DNA sequence. This is the current focus of attention. Here are some results of our application of the third generation sequencing, nanopore. First, we developed a process based on the nanopore long sequence to analyze the genetic background of ARG, identify the host. The result obtained not only give us a new understanding of MGEs, such as plasmic and integrative and conjugative elements, ICEs, but also provide an important reference to further analyze the development of resistant bacteria and the transfer mechanism of ARG in the environment. A total of 10 potential resistant pathogens were detected in our research. Most of the pathogens carry multiple resistant genes, covering 6 types of resistance. By tracking and monitoring this potentially pathogenic bacterial group, we found that 5 of them were present in both influent and effluent of sewage, not being completely removed. Resistance genes associated with MGE, mobile genetic elements, have higher HDT potential, horizontal gene transfer potential, and then high risk than those on the chromosome. Our results shows that most of the ARGs in the wastewater treatment plants are carried by plasmic and integrative and conjugative elements, so-called ICE. The ICE carry 13 subtypes of ARGs, indicating that ICE may play an important role in the spread of ARG. 23 ARG subtypes carried by plasmic existed in the influent and effluent of wastewater treatment plant of Hong Kong, indicating that this mobile and risky ARG were not completely removed and could be an important source of ARG in the receiving water. Thus, we should further strengthen the wastewater disinfection treatment. With the wide application of the high super sequencing, the whole genome of different bacteria is potentially increased in a public database. Deep mining of this big genomic data and machine learning could help us reshape our knowledge of HGT, horizontal gene transfer, and code selection of ARTs and other resistant genes, and guide our analysis and interpretation of environmental metagenomic dataset. Here, I would like to share 3 projects of us for your reference. In the environmental field, HDT Horizon Transfer is a major mechanism of resistant generation and got a lot of attention. Plasmid play important role in HDT, but the previous studies on ART associated with plasmid were mainly based on the plasmid of pure culture. This limited our knowledge. In addition, little is known about the interaction between the plasmid and other mobile genetic elements, such as insertion sequence, so-called IS. We developed a tool for plasmid classification, ART annotation, and visualization, and then use this tool to systematically analyze a large number of bacterial plasmid and the genome. Our result shows that most of the ARTs carried by plasmid closely relate to the conjugated plasmid, and there are also a large number of ART transfer network associated with the insertion sequence, IS, indicating the significant role of the joint interaction between the IS and the conjugated plasmid as the universal mechanism of HDT. In addition to this in silico analysis, we also verified the interaction of the IS and the plasmid and the important role in ART horizon transfer by some laboratory experiment. Another example is the type 1 integron, which is an important ART carrier. It is almost 100% distributed in the gamma class of the Protobacteria phylum. Based on our analysis of 120,000 bacterial genome in the public database, especially they enriched in those bacterial families in the human gut microbiome. The 8 categories ARTs on the type 1 integron are related to the aminoglycoside, beta-lactam, and so on. We also analyzed abundance and the genetic association between ARTs and metal-resistant genes based on the big data analysis of bacterial genome. The results show that the bacteria related to human have more obvious co-selection of ARTs and metal-resistant genes. And the relation between the ART and the metal-resistant genes is also much stronger in the pathogenic bacteria. This explains the co-selection of ART due to the metal pollution in the environment at the genetic level. Of course, this in silico big data analysis need to be further validated by the experiment and the real environmental data set. The source of ARTs in the environment mainly comes from hospital, pharmaceutical factory, wastewater treatment plant, and animal farm. Then a question to ask is, could we use metagenome measure as the management tool to determine the source of ART contamination in an environment sample? In other words, is it possible to track the pollution source of ART? We combined the big data analysis of ART with machine learning approach and propose a new method to track the source of ART. First, we performed feature analysis of 650 global metagenomic dataset of human feces, animal feces, and wastewater natural environment, using the random forest algorithm to establish the predictive model and then configure the samples and adjust the parameter to optimize the model and use simulation data for validation. After that, we conduct MST, microbial source tracking analysis for the real environment sample in Hong Kong and Pearl River Estuary and South China Sea. For 5 of the 12 Hong Kong marine sediment sample, the contamination of ART from wastewater exceeded 10%. Such result match the pollution level of this sample very well. In our another study, a large portion of ARG in the sediment of 2 sampling sites at the Pearl River estuary came from wastewater. With most studies on ARGs, people have realized that risk assessment of ARG in the environment is more important than just recording the concentration and the distribution. There are 2 major types of risk of resistant genes. The first one is the risk of antibiotic treatment failure in the medical field, in the hospital, and the other is the risk of the spread and transfer of resistant genes in the environment. Currently, there is no widely used model for environmental risk assessment of ARGs. In a recent paper, we propose an assessment framework based on 3 major factors, including whether the ARG is enriched in an anthropogenic environment, HDT potential of this ARG, horizon gene transfer potential of the ARG, and also the third one is whether the ARG is carried by the pathogen. This framework proposed the basic risk classification and also provide a practical measure to identify those high-risk ARGs and take the first step to move forward from the surveillance to risk assessment. Within this framework, based on the big data analysis of a large collection of both genome of bacterial culture, metagenome of environmental sample, we proposed a draft list of ARG to be controlled with high priority in the environment. Of course, this list could be further periodically updated when there are more bacterial genome and more environmental data set and more knowledge about HDT, horizon gene transfer, and MGE, mobile genetic elements. Finally, I hope this could be the take-home message. Metagenomic measures are a powerful approach for surveillance of ARG in environment to understand the genetic mechanism and environmental driving factor, and for the development of management tools and also for the assessment of risk. At the end, I would like to thank the hardworking team, the collaborators, and the funding agencies. Thank you for your listening. Thank you.
Thank you, Professor Zhang. Is there any question that from the audience? So Professor Zhang, may I ask one question?
Sure.
Yeah. So I'm not very familiar with the ARG. I used to have some strange ideas. So I take a wrap of my dog, take this tissue for running antibiotics. And I did find several kinds of different antibiotics. And the thing is that I never use these kind of drugs, either in my room or any member of my family use. So could you suggest where those antibiotics could come from?
You mean antibiotics or you mean antibiotic resistant genes?
Antibiotics.
Okay, you mean there are some residue of antibiotics?
Yeah, very strange.
In the sample of your dog? In a sample of your dog? Yeah. Okay. Did you check the dog food?
I did. They are not originated from dog food or dog drugs. No, they are human antibiotics.
So I mean, did you take some sample and for the analysis, using dog food as the sample for the analysis to see whether there are some antibiotics already in the dog food?
Yeah, I did, but nothing in the dog food.
Okay, so that's not the source.
Yeah, food is not the source. Yeah.
Then I do not know what could be the source. You know, if they're not from the food, and also you didn't use antibiotics. for a dog, then I really do not know where they come from. If you
Yeah,
talk
yeah.
about the ARGs, then they could be carried by pathogen, by bacteria. Then that's easy to some other transfer exposure pathway. But for the antibiotics, the chemical, if not from the food, and also you didn't use that, not in the water, then we don't know where it going to come from, because most of them they will not be transferred in the air. Yeah.
Yeah, probably very difficult, right?
Yeah, no possibility like this.
Okay, yeah, skip very strange. Yeah. Thank you for your answer for sure. Yeah. Okay. Yeah, the antibiotics and ARG could be very interesting topic. Yeah. So our next speaker is Professor Jiang Wen Chu. Professor Chu is a professor in Department of Biology, Hong Kong Baptist University. His main research area includes deep sea biodiversity, genetic connectivity, and genome level adaptation to extreme environmental conditions. Today, he will present stress responses in corals, winners or losers under climate change. Professor Chu. Yeah. Great. You've already shared. May play your slide at full mode.
Thank you, Chair. Can you guys hear
Professor
me?
Chu, you are using the presenter mode, so we can see every like next slide. Do you use the full
I'll
screen?
change. How do I?
Probably you should end the sharing first. End your sharing first. Oh,
let's see.
swap. Yeah. We try this one. Yeah.
So this,
Yeah. Good. Yeah, we are good. Yeah.
Okay. Thank you, Chair. For the introduction, I do deep sea research, and but I'm interested in shallow water research as well. So in recent years, I have been studying coral health, especially predation on corals by sea urchins, and as well as corals' responses to bleaching. So we conduct field surveys and we conduct laboratory experiments. Today, I would like to report our work on bleaching, coral bleaching. So for those from Southeast Asia, I'm sure you guys know better corals than we do, because you have more well-developed coral communities. Reef corals, they are distributed throughout the tropical and tropical oceans, and they support high biodiversity. In South China Sea, we have more than 500 species of hard corals, and they are very important ecologically and economically. However, the status of coral reefs worldwide is not in a good situation. So over the last 50 years, there has been more than 50 years, there has been a decline of global coral coverage by more than 50%, and fisheries by 60%, and over 63% of coral reefs biodiversity have declined. According study of more than 3,000 reefs around the world, there has been dramatic changes of decline of coral baselines over the last several decades. So with the predicted increase of sea temperature by this century, it is predicted more coral deaths will result in the future. So I'm sure you guys all know about this coral and Symbolism relationships. They are the foundation of this high productivity in the coral communities. So this symbiosis is very important for the normal function of corals. However, environmental stressors such as temperature, high temperature, low temperature, hyposalinity, high light intensity, pollution, and sedimentation all have been known to cause coral bleaching, resulting in the breakdown of symbiosis. So there has been several large-scale coral bleaching events since the 1980s. So the most recent one is the 2014 to 2017 global bleaching event, which has affected many coral communities. So I'll focus on the bleaching of Hong Kong corals. So I'll briefly introduce the environmental settings and the 2017 bleaching event we experienced in Hong Kong, and also the implications for the fringing communities in Hong Kong, fringing coral communities. And also, I would like to share with you our follow-up studies. So I'll focus on reporting this bleaching event. First, Hong Kong is slightly south to the tropical of Cancer. So the climate is subtropical instead of tropical. So we have great seasonality with a wet season where temperature can be, water temperature can be 25 to 29 degrees, which is ideal, I think, for coral community development. However, in winter, in the dry season, temperature can drop down to 14 degrees. So during the cold season, you don't expect corals to grow. So they are under marginal environment for coral development. So in Hong Kong, we don't have true reefs, but we do have 84 species of hard corals. So this is a typical situation for the fringing communities of the northern South China Sea, compared to Taiwan, which has roughly 300 species of corals. Our diversity is relatively low. Nevertheless, we can find some corals, such as petit dryer, going to a height of 2 meters above a rocky substrate. So in some places, we still see relatively good coverage of corals up to 70%, 80% substrate cover. Coral bleaching has not been a serious problem in Hong Kong due to the subtropical climate. So as we would not expect this kind of environment to experience heavy coral bleaching. So there has been only a few anecdotal reports, not very detailed reports, since the 1980s. And most of these damages caused by bleaching were relatively local, very small scale, not regional scale, although there's been a massive bleaching event in 1997 during this strong El Nino year. But this is, again, this is not a whole Hong Kong event. In several locations, the impact was quite high. But most of the other sites were not heavily affected. And also in 2014, there was a small scale bleaching event affecting 8 sites. So we conduct a field survey, and we also looked at the recovery, and we found the recovery was relatively good, with more than 90% of the colonies we tagged fully recover after several months. So what I'm going to report is a breaching event discovered in 2017 when we undertook a project with a number of collaborators in different universities, including collaborators in CTU, Dr. Leo Chan, who is co-chairing this meeting. So we surveyed 33 major coral communities in Hong Kong. And during the summer, we found these 6 sites with breach corals. So when we discover this coral breaching, we conduct video transfer surveys. Basically, we took videos in the field to reduce the time spent in the field. So after we got the videos, we analyzed these videos in the laboratory using 2 methods. The first method is to use the 5-point sampling method, which means we play the video on the computer screen and we stop the video at a certain time period, and then we overlay the computer screen with several red dots, and we determine the substrate under each dot. For example, whether it's a breaching coral, whether it's sand, whether it's mud, etc. For corals, we determine whether it's healthy or breach. So that's one method. And another method is that we count all the corals that can be found, all the coral colonies that can be found on the transect. Most of these are larger than 10 cm in diameter. We identify them to genus level, and we score each colony as breach or healthy. So just to show you some of the most commonly affected corals. So this is a Pachydrala under bleaching, and as you can see, this Pavola is not breached. And in this photo, the Pavola is breached, and this is Echopora, and this is Varieties, and these are all very common genera of corals in our areas as well as in South China Sea, I believe. So we found coral breaching in this 6 sites, and affected corals vary from 5.9% to close to 60% of coral cover. So for instance, at this area 1, this several genera, Pachydrala, Varieties, and Echopora, they were responsible for 25%, roughly 25% for these 2, and then 2.4 for this Echopora. Breaching. And then for Barazai, this Echopora and Montipora, these are prey corals. They were responsible for most of the breach area. So as you can see, basically several coral genera, they are responsible for most of the breaching we discover in Hong Kong. And only 6 of the sites had corals undergoing breaching among the 33 major coral communities we surveyed. So that's the 5-point method. Using the colony count method, we found a similar result. So we found roughly 20 to 56% of colony breach in this 6 sites. And in Barazai, where the prey corals are are dominant. The breaching was most serious, and at the coral breach inside Hoi Ha Wan Marine Park, most of the breach came from Pavona. So that's the observation of the field situation. After this observation, initial observation, we conduct tagging. So we tagged roughly 200 colonies, less than 200. And for each breach colonies, we use a steel bar with label to tag the colony. And after 1 month and after 3 months, we went back to the site to check the recovery to see whether they have died or they have recovered. So this is the situation. We tagged Pavona, Petidrella, Goriopola, Turbinaria, Ecopola, Politis, and Lepistria. These were all the coral colonies that had suffered from breaching. And then after 1 and 3 months, we went back to check the recovery. So this shows the recovery of this different genera of corals. So overall, we see most of them have, most of the coral colonies had a good recovery of close to or over 90%. Only Goniopola had a relatively low recovery rate, and that's because probably due to the few colonies available for tagging. But overall, as you can see, the recovery was quite good. So we tagged roughly close to 200, but we only recover 137 in the follow-up surveys. Among this, 80% have fully recovered, and roughly 20 have partially recovered, and roughly 3 had suffered from mortality, which means they died after breaching. And noticeably, those that eventually die, they are mainly ecopola and some multipolar. So these are pre-corals. So just look at the before and after photo. Before and after photos of this coral colonies. So this is a Petidrella before and after recovery, and Politis as shown over here, and then as shown over here, recover. And Ecopola breach colony, and then over here a recovered colony, and Pavona as well. So these are partially recovered colonies. So as you can see, this was a breach colony, but only a small patch of the coral remained, but this is fully recovered, all right, this part. But the other sections were missing after breaching. So after they die, they probably become very fragile. And also the same for this colony over here. Only this part recover, and this part is dead. So we would like to know what factors trigger the breaching after observing this phenomenon of breaching and recovery. We didn't have environmental sensors at this size, but in Hong Kong, Environmental Protection Department had a network of environmental monitoring stations. We chose one that is relatively close to our site, and we looked at several environmental parameters: salinity, chlorophyll, and DO, and temperature. we compiled the data from 2007 to 2016 on this side, and the 2017 data on the right-hand side. As you can see, we didn't see much changes in the chlorophyll. So we looked at the changes in different months. We didn't see much changes in the chlorophyll, much differences between this particular year, 2017, and 2007 to 16. We didn't see much differences in chlorophyll, DO, and temperature, but we saw a lot of differences during the summer. Surface salinity dropped from roughly 30 parts per thousand to close to 15. That is a lot of fresh water. So there was heavy rain before the salinity drop was recorded. And as you can see, the data is not very dense. So every month there's only one data. So we might not have captured the lowest salinity happened during this particular year and before the discovery of coral bleaching. So this led us to conclude that maybe salinity was the culprit. Low salinity was the culprit of this particular event. So from our study, we found a very mild impact of coral bleaching. However, if you recall, in 2014, we had a similar small-scale impact. And in the future, based on the prediction of the Hong Kong government, there may be more extreme rainfall, extreme temperature event, high temperature events. So this kind of cumulative impact may not, should not be ignored. And also, we found that there's selective bleaching of several coral species, as well as selective mortalities, especially the prey-like or ecopola, as well as some multipolar colonies. So this kind of cumulative impact should not be ignored because they have the potential to shift the community from a more diverse community to a more monotonous community dominated by, for instance, Pavona or the varieties. So we feel that in the future, if this kind of events happen again and again, there could be winners, losers, and losers in among the existing corals of Hong Kong. So this is the field study. But during this field study, we also collect some samples. We collect the healthy sample, we collect the normal sample, normal looking sample in a breach colony, and we also look at the breach. We also collect the breach tissue, and we also look at the recovery tissue. So we collect this kind of tissues. We also run transcriptome analysis to determine the pattern and in gene expression to look at what happened at the metabolic level, at the immunity level. So recently we have published a paper on the host and symbol transcriptome responses during the natural breaching and recovery in Pavona. And we are analyzing some more data on Pachydryla as well as Montipola. These are very different species. And also they are different in their responses to breaching as well as the recovery, to look at whether their metabolism are different, whether their immune system responses are different, and whether they are under different levels of oxidative stress. And also with the support of UGC Hong Kong, as well as ECF, Environmental Conservation Fund, we have established a laboratory system. This is one section of the system where we have individual tanks that allow us to do laboratory culture of corals and conduct laboratory experiments to determine temperature and salinity stressors to corals. So we measure the algal concentration, we measure chlorophyll, we measure also transcriptome of the host and symbol, and we also measure the bacterial composition to see how this kind of different patterns of the symbiosis are broken and doing a breaching. And also we use 9 species of corals to do this kind of experiments. We have just complete this kind of experiments. We haven't got time to do the comparison, but our initial results shows that branching corals, they are very susceptible to temperature, high temperature, but all of the corals, they are very resistant to low salinity. We use a low salinity down to 21 parts per thousand, but most of these local corals, they can survive under this kind of low salinity conditions for up to more than 1 month. So that's very surprising. So I hope in the future, I will, in the near future, I'll be able to report the results. So also with this kind of study, we can gain some insights into how different molecular pathways are involved in their differential sensitivity, underlying their potential fate in face of global environmental changes. So that's all I would like to report. I would like to thank the funding bodies as well as my lab members who have contributed to this work, Yanjie, James, and Yolanda and Jack. Thank you very much for your attention. So
yeah, thank you, Professor Chu. Is there any question from the audience? Oh, actually, we do have a question from Professor Cabansat from University of Philippines, Visayas Tacloban College. And the question is, there are already studies focusing on high temperature resistant species. Would you recommend these species to be transplanted to reefs adversely affected by bleaching? Why or not?
That's yeah, I have read some papers considering the transplant of high temperature resistant species. But in Hong Kong, we are in the beginning to understand which species may be more susceptible to temperature and salinity stressors, and which one may be very resistant. I hope after we discover this kind of phenomenon, we can consider this in the future transplantation experiments. Currently, I think the restoration program is rather cowboy style. I think they only consider which species grows faster, and they tend to use ecopora. And in our experience, all species of ecopora we test in Hong Kong, they are very susceptible to high temperature. So if you don't consider this temperature susceptibility for a restoration program, then there's a high likelihood that your project would not be successful. So thank you. I'm not sure if I have answered your question.
Yeah, thank you very much. Yeah, I do have a follow-up question. So besides the temperature resistance, do we need to consider other factors like the suitability of the coral species? I'm not very familiar with this area, but
yes,
probably,
Chair. I think we only look at the phenomenon right now, but actually there could be a genetic basis for their differential temperature resistance as well. And also in this coral transplant restoration program, we also need to consider the genetic background.
Sure. We have another question. Have you assessed if previously bleached recorded corals have same reproductive capacity as unbleached?
Well, this is a very good question. We haven't had the opportunity to look at their reproduction, but I hope we are considering this collaboration with a colleague in Chinese U, Dr. Abel Tree, who has conducted some coral reproductive experiments. So hopefully we will have data. I think their reproduction must be compromised, but we don't have data. Thank you.
Thank you very much. We have probably the last question due to the time issue. So another question will be from the presentation. The recovery rates were noted after 2 to 3 months. How is this compared to recovery rates from other areas?
I think will be comparable. Most in depends on the if the condition improves, some corals may be able to recover. But if the condition, the worst condition, the worst environmental conditions persist, then corals may die. So maybe quite difficult to compare recovery rate from one location to another because the situation may be different. So you need to look at the environmental conditions during and after bleaching. So we were quite lucky. Probably in our case, the salinity, low salinity event did not last very long.
Okay, thank you very much.
Thank you, Chair.
Yeah, thank you.
And thank you for the audience. So I stop sharing.
Yes, please.
Okay.
So our next speaker is Dr. Leo Chang. So I've already introduced Dr. Chang. So his presentation topic today will be the recent progress and future research on Cicoteria causing marine benthic dinoflagellates in Asian Pacific regions.
Yeah, thank you, Henry. So
thank you.
You can see my PowerPoint, right?
Yes.
So no problem, right?
Yeah.
Yeah, so I don't need to introduce myself. So I will go to my presentation. So first of all, what is Cicoteria fish poisoning? I think for most of the friends come from Southeast Asia, they may know that. So Cicoteria fish poisoning is a foodborne illness caused by the presence of cicotoxins in the flesh and visceral of tropical and subtropical coral reef fish. More than 400 species of bony fish at all trophic levels have been identified as cicotoxic. Cicotoxin is produced mainly by dinoflagellates that are usually attached on macroalgae, sand, coral rubbles, and seagrass in the sea. especially in tropical areas. That means the Southeast Asia. Organisms in the group of benthic dinoflagellae have been implicated as the causal organism of CMP, such as Porocentral, Curia, Ostratus, Gambidisca, Fukuyu, and Aphidenia. However, many of these species have been reported to produce various types of toxin, but it is believed that only toxin from Gambidiscus and its closely related species Fukuyu reveal the symptomology corresponding to the ciguatera fish poisoning illness. So this is the SEM and then the live photo of Gambidiscus. So they are sized just as the diameter of a hair. So in fact, if you have good eyesight, you can see them under the sea. So there are more than 18 species in the Gambidisca and Fukuyu families. They produce gambiotoxins and a suite of ciguatoxins. So what is ciguatoxins? Ciguatoxins produced by dinoflagellates, right? I already mentioned that from Gambidiscus and Fukuyu. So more than 50 analogs of CTS has been identified. So they are polycyclic eaters. So they have a leather-like structure. They are very stable, resistant to heating and freezing. So you cannot get rid of them in the fish. So they also resist mild, acidic, and basic conditions. And they are lipophilic, higher concentrated in fatty acids. So if you intake the ciguatoxin, they will stay in your body forever. So from our many years study, we start the whole footwear study from Gambidiscus, and then to herbivore fish, to carnivore fish, and then to seafood consumers. But today I only will focus on the benthic dinoflagellates. So at the beginning, it's very difficult to get the ciguatoxin fishes. So I went to the Republic of Kiribati and sample along the most endemic area, and then we have ciguatoxin concentration with the location. So after we get this map, then we dive into the sea, and then we try to isolate dinoflagellates. So in fact, these fish are our detective. So let us know how the distribution, abundance, and density of Gambidiscus in the Republic of Kiribati, mainly the western side of the island. So from the trip, we isolate a lot of Gambidisca highly toxic species, and you can see this species is very toxic compared to other species. So in collaboration of Professor Choi Peng Liu and then Ludo Ding from Malaysia and China respectively, we identify a new Gambidisca species type 6 as the Gambidisca balachi. So most of my students are now working on this species. So you can see the ciguatera is an issue of local, regional, and global importance. In the past, CIP was only endemic in the tropical and subtropical region between latitude 35 degrees north and 35 degrees south. And ciguatoxin, according to their distinct structure and geographical distribution, are classified into 3 groups: Caribbean ciguatoxin, Indian ciguatoxin, Pacific ciguatoxin. So this make the ciguatoxin research more challenging because they have a diverse structure and toxicity. So now there's widespread of ciguatera fish caught from the endemic region to other parts of the world because of the high demand for coral reef fish for seafood. So which lead to the inter-regional trade of large volume of these fishes into different areas around the world. CIP so now become increasingly prevalent with the effect of globalization as well as the climate change. So now the ciguatoxin is widespread everywhere. So you can see this is the global distribution of ciguatera fish poisoning. So global warming may also lead to an increase in CFP cases. So an observable positive relationship between CFP incidence and average sea surface temperature has been discovered. Why? Because the temperature is the primary environmental factor regulating growth. Can be discussed tends to growth at better at higher temperature. And Professor Chu already mentioned there are global bleaching due to global warming. And in fact, more coral bleaching provide more surface for the proliferation of these toxic can be discussed species because they need to attach to those turf, grow on those bleached coral or dead coral. So this is a study by Marina China, my collaborator in French Polynesia, and she discovered that coral mobility may be the factor in the genesis of natural occurring growth of Gambidiscus by providing more new surface for colonization by this opportunity macroalgae that are ideal hosts for Gambidiscus. So these observations support the hypothesis that CFP outbreak can occur in response to disruption of reef. So more disruption, more toxic fish. So in the future. So the time lag may be only a few months for them to grow. So you can see that the hot season, the coral bleaching, and then later you find the high maximum cell abundance and toxic production found in this area by the Gambidiscus toxicus. So my collaborator, Professor Lin Boti and Cui-Ping Liu also find that this disturbance that alter benthic substrate type can have profound effect on the composition of species present. The highest Gambidiscus cell abundance was detected in the microhabitat dominated by algal turf associated with coral rubble fragments. And this is the summary, the climate change, local threat, local stress, like overfishing, pollutant, will cause disturbance to the healthy coral reef. And then this degraded coral reef, they will be the best surface to host these beta species, the benthic dinoflagellates. And then their toxicity will cause the shift of the alteration of the trophic transfer, and then will change the species diversity in that coral reef. So don't underestimate the ability this tiny dinoflagellate to the whole coral reef ecosystem. So this is my prediction 10 years ago. The number of CFP may further increase because the overexploitation of coral reef fish, that continued degradation of coral reef due to global climate change and other anthropogenic disturbances, as well as the expanded distribution and elevated abundance of toxigenic Gambidiscus globally. So my prediction is that cicadas fish entering market around the world is going to increase. So in 2017, my collaborator, Professor Lin Boti and Cui-Ping Liu, and also my Vietnam collaborator, With Hao, they already discovered some local fish, the snapper, are identified contaminated with cicatoxin. So this is not import from other area. This is a local find. And also, Dr. Tao from Vietnam, the Institute of Oceanography has carried continuous surveillance on the CFP, and she discovered from the 2008 to 2018, within these 10 years, there are many local CFP incidents has been identified. And mainly from the red snapper and the moray eel. And she found that CTH1B is responsible for snapper poisoning in Vietnam. So this is the map showing the Pacific region, the distribution of Gambidiscus species. So after literature review, we find that there, in fact, a lot of Gambidiscus identification in Japan, Korea, Taiwan, Okinawa, China, Hainan, Vietnam, Malaysia, Indonesia, etc. And then in Japan, the Okinawa has found different kind of Gambidiscus species. And Professor Lui Songhui from Chilean University has conducted systematic sampling, and he found that 3 species has identified as Gambidiscus in Chinese waters. So, so we are able to confirm some Gambidiscus from Malaysia and Kiribati, and with several new records in collaboration with Professor Trepe Liu and Lin Bo-Tin. So in response to the ciguatera fish poisoning in Vietnam, we actually lead our research to go to conduct some benefit dinoflagellate sampling in Vietnam, and we discover there are many Gambidiscus species and other benefit dinoflagellate in the degraded coral reef. And in collaboration with international academia, especially I think from the Southeast Asia, the Vietnam, Malaysia, Indonesia, we will conduct a future research to investigate the bioactive potential of benefit and epiphytic toxic dinoflagellate and their associated microbiotas. So you can see that there are many people research on pelagic harmful algal blooms, but there are still not many scientists working on benefit and epiphytic toxic algae. Unlike planktonic microalgae blooms occur often in the upper part of water column, so you can see them. There's no visible environmental phenomenon such as blooms and red tide can be easily linked to benefit dinoflagellate, and then outbreaks are often overlooked. So this is when we go to study the benefit dinoflagellate sampling, we always observe this kind of underwater blooming just above the coral and cause coral bleaching, massive death of invertebrates. So we develop our unique sample procedures. We design some kind of in situ auto sampler we can bring down to sample the target dinoflagellate. And this is a short video showing our so everyone can see the video. So we use this sampler to conduct our trial survey sampling in Hong Kong November 5th, and then we find 5 species of this benefit dinoflagellate species. So the identification are still ongoing. And once we get this single cell, we can mass culture to study their toxicity and other kind of applications. So we will develop into 2 kind of SOP. First is purification of the toxin, and then the second part is species identification. And eventually, we hope that from this dinoflagellate, we can produce a low price toxin to support high impact biomedical pharmaceutical research. So this is just a brief summary of our recent research. So at the beginning, we start the ciguatera research for food safety monitoring. And then we purify 74 micrograms of cigatosine from the moray eel, and it costs 1.73 million. So the toxin standard is very expensive, $3,000 per 1 microgram. And then we developed 2 rapid, robust, and reliable analytical methods for toxin analysis. And then now we move on to the identification and characterization of bioactive secondary metabolite from those bioactive dinoflagellates. So use molecular networking to identify new cigatosine analogs. As well for drug development, we also use the molecular networking to isolate secondary metabolite for pharmaceutical applications, such as the control sensing anti-inducer and inhibitor discovery from marine microalgae derived bacteria. And also, most of people focus on cigatosine because they are lipophilic, so they will transfer along the food web, but not much research are on metotoxin. So our research also focused on the metotoxin. They are water soluble. Firstly, we discovered 7 super carbon chain compounds, and more than 10 metotoxin analogs were discovered, and their structure formula were now elucidated by MS spectrum. So why this is exciting? Because most of CTS analogs are new compounds, and these macromolecules are not protein-based molecules, which make this discovery very exciting and meaningful. So I just show you some of our new discovery metotoxin macromolecule. Some are known compounds, some are new compounds, and some. So this is all exciting results from this. So based on our 10 years research experience, we also provide training workshop and introduction of scientific diving for marine dinoflagellate sampling and processing. So we conduct a training workshop in September 2018 for UNESCO Westpac. And now, thank you for your attention and acknowledge the funding organization. Thank you.
Thank you very much. Thanks, Leo. So before our break, ladies and gentlemen, would you mind turning on your camera so we can have a group picture again for all the participants? Thank you very much. So yeah, great. So Grace?
Yep.
Yeah, would you mind take the pictures?
Of course. So let's give, say 15 more seconds for people to turn on their cameras and get ready, and then we can take the photo.
Great.
All right, so as usual, I'll also take a few. So on count of 3, 3, 2, 1, cheers. Okay, and another one. 3, 2, 1, cheers. Well, because we have a few more participants, so I'll have to take 2 more photos. Okay,
Yeah,
sorry.
sure.
3, 2, 1, cheers. And one final one. Ready? 3, 2, 1, cheers. Okay, thank you so much.
Okay, so, sorry, we have a question actually for Leo. Yeah, so the question is, in the late 1990s, I've been worked with Dr. Fukuyo and Dr. Marisingat isolating ciguatera benthic dinoflagellates, which we've deposed in central Philippines, Seguros and Reef. So is it possible to collaborate? So that's an offer for collaboration for Leo.
Yes, Francis, certainly, I think this is the purpose of today's symposium. And we want to share our latest research, and then we also look for collaboration, especially in Philippines, because we have a collaborator in Thailand, in Malaysia, in Indonesia, and we don't have a very solid collaborator in Philippines. So definitely, we are very pleased to collaborate with your team or other Philippine researcher. As you can see that we train people along our sampling. So whenever we go to Vietnam, we go to Thailand, we will train local scientists. And also we will try to develop some kind of international standard for this kind of sampling method for comparison, comparative study and collaboration. So thank you for your kind offer.
Yeah, we have several raising hands. So how can I open up? So do we need to open up? So Dr. Jingling, do you have any question?
Yeah, Leo, thanks for an inspiring talk. So your current focus is on the benthic and epiphytic, right? So I'm wondering, is there any dynamic interaction between the pelagic and the benthic system in terms of those algae and the algal toxin, those things? Yeah, thank you very much.
Yeah, I think those benthic dinoflagellate have some kind of stage they will become pelagic.
Okay.
So I think, and also they also secrete this lipid philip toxin into the water column. So this toxin will be traffic transfer to zooplankton, etc.
Okay. So they are not strictly isolated. So there are still some dynamic interactions.
Yes, still some kind of linkage.
Okay. Okay.
Yeah,
Good.
but they prefer benthic, right? Yeah, but sometimes maybe if the condition is not good enough, so this is why you see those dinoflagellate can transport to other part of the world through the water current or the ballast water.
Okay. Okay. Thank you. Thank you very much.
Yeah. Thanks, Nathan. Lenny, do you have anything to say?
Yeah, I just want to say that there is already, and we actually find many benthic diatoms and dinoflagellates related to ciguatera shellfish poisoning, but not very. Since the Philippines is mostly focused, and the big problem here in the Philippines is paralytic shellfish poisoning caused by Pyridinium bahamense. That's why the focus of most of the researchers in harmful algal blooms are in Pyridinium, but we do see many of these in our samples, but not so many people working on it. Although there was one research before in Bolinao working on Gambierdis, because they set up a mesh in the bottom so that it can collect it can attach that mesh there in the bottom so they can collect those kinds of dinoflagellates. So yeah, it's a good actually, since there are not so many people working in the Philippines on Gambierdis.
Yes, I'm not deliberately left out Philippines. I try to link up with Philippine researcher in a harmful algal bloom many times. I approach Professor Rajanda, right? And then it seems that all of them are interested in pelagic research, like the paralytic shellfish poisoning, DSP, etc. Right? Not many people are interested.
Yes, because that's the main problem. Yeah, and paralytic shellfish poisoning is just really causing deaths here, and that's why
you see those kind of PSP or DSP, they are water soluble. So in fact, yes, if you use depuration, they will be cleaned off.
Depurated, but
will be more problematic in seafood poisoning in the future.
Yeah, but there is still until now deaths in paralytic shellfish poisoning, and the system is not still very good. How the early warning system, like the warning system. So that's why many are focused on that. But yeah, I agree that we should not forget other harmful algal species that are there, that we also have here in the Philippines. There are a lot, and even in Ciguatera fish poisoning, there was actually a case in Iloilo of Ciguatera fish poisoning already, but again, nobody is pursuing this area.
Yeah, we will be very pleased to collaborate with Philippine scientists, and then we can go there and then sample with you and then collaborate. So I go to Philippines for diving training many times, but only for training, not for scientific research. Yeah, so please keep in touch and email us if you are need our help, because I think in the regional.
Yeah, we would. Actually, I know Dr. Lim Putin and Dr. Chin Liao, so I thought already covered in that area. But if you are looking for other samples in the Philippines,
yeah, definitely. We are eager, looking forward. Yeah.
Yes.
Yeah. Thank you. Yeah, Lily.
Yeah, thanks, Lenny. Probably we will have only 1 minute short breaks. So we will come back in 1 or 2 minutes, and Dr. Leo Chan will be the following chair for the remaining session. So I will see you pretty soon.